Transmission lines, nerves and the structure of them, and the repair. And the repair might surprise you, but in the peripheral nervous system, we can get some repair depending on what part of the nerve is damaged. So let's start with nerves. And remember, we said groups of cell bodies in the peripheral nervous system would be called ganglia. So associated ganglia. A nerve is a cord-like organ. It's found in the peripheral nervous system. It's a bundle of myelinated and non-myelinated peripheral axons enclosed by connective tissue. There are two basic types of nerve depending on where they originate from. There are those that come off the spine called spinal nerves, and the cranium called cranial nerves. So that structure of that simple organ we're going to call a nerve is going to have names that are very similar to what we saw in muscles. Around each axon and its myelin sheaths, there are, and those myelin sheaths are made by Schwann cells in the periphery, there's a loose connective tissue that's called the endoneurium. Coarse connective tissues that bundle a group of fibers into fascicles are called perineurium. And an epineurium is a very tough fibrous sheath that is around all of those fascicles, and that forms the nerve. So these structures should be reminiscent of what we saw in the organization of muscles. So here we see a nerve. You can see endoneurium around each of these individuals. Those are grouped together in a fascicle. Whoops. Here's a better, clearer one. And then all of these fascicles are grouped around with epineurium on the outside. In an artist rendering, individual endoneurium around an axon, groups of those brought together in perineurium, and outside we have the epineurium, and also in there are other tissues such as blood vessels. So we can structurally classify them. Most nerves are going to be mixtures of afferent and efferent fibers. So they're going to have some axons sending information toward the central nervous system and some sending information away from the central nervous system. They're also going to be a combination of somatic and autonomic fibers. So we can classify them based on the direction they transmit impulses. And mixed nerves would have both sensory and motor fibers. They would be carrying information to and from the central nervous system. Others would be sensory or afferent nerves. They are only carrying information towards the central nervous system. And others would be motor. They are carrying only impulses away from the central nervous system. Pure sensory or pure motor nerves are rare. Most of them are mixed. We will see some pure sensory when we look at the cranial nerves, but most are mixed. So the two types of fibers in mixed nerves would be somatic afferent, somatic efferent, visceral afferent, and visceral efferent. The somatic afferent are taking our sensory, then going from muscles to brain. Somatic are motor from brain to muscle. Visceral are from sensory organs to the brain, and visceral efferent motor from brain to organs. And a ganglia, again, is a collection of neuron cell bodies. They are associated with nerves in the peripheral nervous system. Ganglia associated with afferent nerves. Fibers have all the cell bodies of sensory neurons. They are found in the dorsal root ganglia, and we talked about those in chapter 12. Actually, we are talking about them in chapter 12 right now. And then there are ganglia associated with efferent nerve fibers that contain autonomic motor neurons. Those would be the autonomic ganglia. And we will cover those when we get to chapter 14. Mature neurons are amitotic, but if the cell body of a damaged nerve is still intact, the peripheral axons can regenerate in the peripheral nervous system. This won't happen in the central nervous system, but in the peripheral nervous system, if you keep the cell body and damage the axon can be repaired. Most of the central nervous system fibers never regenerate. The oligodendrocytes have growth inhibiting proteins that prevent regeneration. Astrocytes are going to form scar tissue in the central nervous system. Uh, there's experimentation happening all the time to see if we can fix some of this, perhaps neutralizing growth inhibitors, blocking receptors for inhibitory proteins, or destroying the scar tissue component. But so far, it has not been extremely successful. In the peripheral nervous system, we can regenerate the axon if the axon fragments in the myelin sheaths distal to the injury degenerate. That is called Wallerian degeneration, and that would spread down the axon. Macrophages would then come in and clean up the dead axon debris. Those Schwann cells that are there would be stimulated to divide. The axon filaments would grow through the regeneration tube formed by the Schwann cells, and as the axon regenerates, new myelin sheaths will form. So if we have damage here, cut the axon, and that ends will seal themselves off, and axon transport is interrupted. That causes the ends to swell. Without access to the cell body, the axon begins to degenerate distal away from the cell body, and that is called Wallerian degeneration. The Schwann cells and macrophages clean out the dead axon distal to the injury. Macrophages will help dispose of the debris, release chemicals that stimulate Schwann cells to divide, and the axon filaments will grow through that tube as the Schwann cells release growth factors expressing cell adhesion molecules. The Schwann cells would line up along the tube of the remaining endoneurium, forming a regeneration tube, and that allows the axon to sprout and continue their original contacts. And the Schwann cells would continue to protect and support that regenerating axon until a new myelin sheath is producing a new myelin sheath. This is a very slow process. It can be